All right, fellas. So this is the second of three videos on DNA replication, and we will get started right now. Now, what this is indicating is the essential. It's a blueprint kind of for how DNA polymerase adds nucleotides onto the template strand. Now notice the template strand in this case is on the right hand side, and so we're starting at the 3' end and we're moving toward the whoopsie daisy, the 5' end. All right, and so this thymine is being added to the adenine here. And the last one that was added was guanine, and the one before that was cytosine, and so on and so on. All right, and so the DNA polymerase then does its job. It removes two phosphate groups from the end of the DATP and essentially adds the thymine to the adenine that's already on the template strand. Now, a little bit more detail on how that might work. I don't know where to hide myself. I'll put myself here. How's that? So this works in what we call an antiparallel elongation format. And so if the origin of replication is right here in the center, so this is the replication bubble, the origin of replication is here. And so 3' to 5' moves this way and this way. All right, so that's what we're going to talk about first. Those are what's known as the leading strands. However, what happens behind the origin of replication? All right, well, and here and here. In that case, we still have to move from 3' to 5', but we're constantly moving away from the 5' end. And so we do this in a slightly different way. Let's concentrate on the leading strand at first. All right, so first of all, DNA polymerases add nucleotides only to the free 3' end of a growing strand, and therefore a new DNA strand can only elongate in the 5' to 3' direction. And so here we are. Here's the 3' end of the template strand. So we add a 5' RNA nucleotide here and move this way. This enzyme right here represents the DNA polymerase, one of the DNA polymerase molecules. Here's the helicase, which is unzipping this way, right? We're moving in this direction. The DNA polymerase is coming behind the helicase and adding DNA nucleotides after the RNA primer has been added by primase, okay? And so this is a continuous direction. So what we're looking at in this position is essentially this right here, okay? And so helicase, binding proteins, and RNA polymerase in this case. So along one template strand of DNA, the DNA polymerase synthesizes the leading strand continuously, moving toward the replication fork. Let's go back to the picture. All right, here's the leading strand, and the DNA polymerase is just adding nucleotides all the way along, following along behind the helicase. Okay, take a minute or two and go back and look at the original PowerPoint for this presentation and watch this animation. It'll show you the differences between leading strands and lagging strands. Now, on the other hand, what do we do with the bottom piece? All right, this piece here. All right, well, in this case, the lagging strand is actually put back together or copied in pieces, not in one single smooth flow. So to elongate the other new strand, which is called the lagging strand, the DNA polymerase must work in the direction away from the replication fork. And so we do this in pieces, and we call these pieces Okazaki fragments. All right, and they are again joined by a molecule, another enzyme, which is known as DNA ligase. So how does this happen? Well, we'll start here. You see the template strand. Here's the RNA primer. All right, it's adding RNA nucleotides, and so we're going to start here. But notice there were already, and there's another primer over here that the primase already glued on. After the primer's been attached, we can then have DNA polymerase form this first fragment, and there it is. And then the DNA polymerase is detached, and it'll end up going back over here to the next RNA primer. All right, and it'll just keep going back and back and back and back. And so on the lagging strand, you're only essentially building small fragments of the new strand at a time. Okay. So here's the next stage in this, and you can see the DNA polymerase has come back and now is working at this point, right? The first one is already finished, but now another DNA polymerase is coming along, and what it's going to do is take these RNA nucleotides and replace them with DNA nucleotides. Notice they're gone in this picture. And then finally, another enzyme called DNA ligase is going to take, you see the little opening right there? All right, the two DNA nucleotides here and here are not actually attached together until DNA ligase ties them together. Okay, and that's the way that this works. All right, so one fragment at a time, and again, worked on by a series of different enzymes. All right, go back, watch this video. Again, the animations are beautiful. Watch this one as well, and this one, my friends. Okay, so to put it all together, on the top here, you can see the leading strand, all right, and this is being put together in one smooth motion, all right, following along behind the helicase. The DNA polymerase is just adding nucleotides as we go. On the other hand, we start with an RNA primase on the lagging strand. Then the DNA polymerase comes in and attaches to the primer. Then another DNA polymerase comes in and replaces the RNA nucleotides with DNA nucleotides.
It's 5 o'clock.
Thank you, Jesus. And finally, the DNA ligase comes in and ties together the new DNA nucleotides. Okay. This is a great little chart found in your textbook, which summarizes the functions of all of the various proteins here. And most of them are enzymes, although there is the binding protein, which is not an enzyme. All right. All right. So what is the DNA replication complex? All right. These are all of the proteins that participate in DNA replication. All right, we call it the DNA replication machine, and this may be stationary during the replication process. Some studies have actually shown very recently that it almost works like a fishing reel, and you just kind of reel in the DNA, and so the complex sticks together and the DNA passes through it. All right. However, the folks are still working out how that might actually work. All right, and so this is a pretty little diagram that shows all of the enzymes in a little bit more complex fashion. All right, but you've got the ligase, the helicase, the primase, the two polymerases, the binding proteins. They're all shown here. Okay, that, my friends, is the end of the second of 3 videos on this topic. All right, stay tuned. I'll get to the next one in just a minute.